Welcome to day 62 and another installment of Neil blabbing to try to help you out, make you smarter. If we look at this, our target mainly is going after digestive enzymes, making sure we know what they do for us, what the subunits are that they break things down into, and then the four major anatomical differences between frogs and humans, because normally you'd be cutting up a poor little Kermit this day, but we don't get to do that. Boo-hoo. Now, other things that we should notice, of course, you would have picked up GR 42 last class. You should have done at least half of it before this. And you're going to need GR number 41, which would be the focus of the first half of this. And then we go after the cardiovascular, the second part of it. Cool. So if we look at this, we should be able to say, hey, I'm going to eat this beautiful pizza and wrap my stoma around it with the Oreo cookies and the Swedish fish and the French fries. Oh, man, it looks good. You should be able to recognize that you got to digest that thing. So the way you're going to do that mostly is enzymes. So the AP is going to ask you things like, what do these enzymes do? Where are they used? What's their function? And you should be able to tell me what part of the pizza they attack, right? You'll notice a lot of them start with like aces and ins. Hey, it ends with in, like protein ends with in, because a lot of them are protein-based, right? Cool. Write down the question, set up the chart, give yourself like a whole maybe 2 pages worth of room, because there's a lot of information that's going into this. And then break it down, see if you can do it. Then pause me. And now we're going to dive into it together, because I don't get to pause anymore. There's this stuff called amylase. Being an ACE, you can recognize that it is an enzyme, a protein-based enzyme, and it targets, as most things in bio tell you, amylose. So, of course, amylose being ose at the end, it is a saccharide. And this one, of course, is the famous name for starch. Now, they're going to call it amylose on the test. Amylase breaks that down. Starch, of course, is a long chain of a whole bunch of sugars. It's a polysaccharide. This breaks it down into what's called a disaccharide. That's 2 sugars stuck together. And that's why if you chew on a nice, stale, boring cracker for a little bit, it'll start to taste sweet because it breaks it down into sucrose. Now, sucrose is still relatively large, and you could possibly pass some of it into your cells, but really you're going to break it down into smaller parts like glucose and fructose. But you can start to taste it with your taste buds. That's why it's showing up. You're starting to chemically digest it. This is happening in your spit, so amylase is produced by the salivary glands, and it's used in your mouth, which is nerdily called the oral cavity. Yeah, you can almost read that, right? This is going to go after the crusty part, the starch section of your pizza, especially if it's got a double crust. Oh, yeah. And then we should recognize that we also use it in another place. It's also produced by the pancreas. Those beautiful scrambled eggs, which you would be seeing inside yourself and inside of a frog, do so many things for us. So when in doubt, on the AP, scrambled eggs, right? If the pancreas is producing it, that means it's going into the duodenum. That means it's being used in the small intestine, where most chemical digestion actually occurs. Now, pepsin. Pepsin, as the name tells you, it ends with an in, which means it's a protein-based enzyme. And that pepsin also, of course, breaks down things called polypeptides. So this is going to break down proteins and turn them into smaller polypeptides. We should recognize that we have these huge globular proteins and functional proteins we take into our body, and we're going to break those down into their chains of a bunch of amino acids together. Now, they're not fully broken down. You still couldn't get those through a cell membrane, so there's going to be other steps later on. Where is pepsin produced? Pepsin is produced by the stomach. And most people recognize, oh, yeah, I got stomach acid. Hydrochloric acid has 2 jobs. One, it sterilizes your food and killing off bacteria, and the other is its job is to activate this thing called pepsinogen, the building block, the inactive form, and turn it into the active form called pepsin. You wouldn't want pepsin active all the time, because then if there was no food in your stomach, you'd start to digest the muscle that your stomach is made of. It's used in the stomach itself because it works best in an acid environment, that storage bag. Now, this is mostly going to be found in stuff like the cheese and the pepperoni, because the pepperoni is made of polypeptides. Kind of makes sense, right? Trypsin and chymotrypsin I put together because they're both produced by the same thing and go after the same target. They're trypsin, which generally goes and is produced by the pancreas. And if it's produced by the pancreas, that squirts into the first section of the small intestine called the duodenum. The duodenum by far is the most important area for chemical digestion in your digestive tract. That means it's going into the small intestine and be used throughout that, right? As it's doing that, it needs the basic environment of the pancreas. That's why it also produces bicarbonate into that small intestine, so that you have a non-acidic area, because that's the pH that's optimal for trypsin and chymotrypsin. Then we can see, of course, this is going to go after proteins again. It's going to go after different proteins and select for those ones. It's also going to turn them into small polypeptides because there's lots of different proteins and enzymes have to have specific shapes to act on. So it's breaking them into their long chains, right, from the big globular things. Once again, it's going to be in the cheese. It's going to be the pepperoni. It's going to be in the ham, right? Then there's these things called peptidases. And hopefully you notice, of course, we're kind of moving our way down the digestive tract. We've gone from the oral cavity into the stomach, and now we've made our way into the small intestine. We're pretty much going to stay there. As we get into that, of course, the small intestine is going to produce this itself in the lining of the small intestine. It's peptidase, so you can guess it's going after polypeptides. It's going to break them into their building block, amino acids. Now, the great thing is now that they're not in chains, in single amino acids can actually fit through a membrane, and you can start to absorb them, which is the other job of the small intestine. That's where you're going to be doing most of the absorption of your actual nutrients that you use in your body, aside from water, right? If we look at this is the pepperoni, this is the cheese, this could be the proteins that are inside of the peppers and stuff that are there too. Now, bile, you'll notice, does not actually have an ACE or an N attached to it because it's not a protein, it's not an enzyme, it's a salt. And salts, of course, are ionic compounds. And when you put them into a solution like your small intestine, they're going to take and dissociate. Now, the cool thing about this is bile is actually produced by the liver, but it's stored by the gallbladder. Part of the reason that structure, that little balloon, has that greenish color. It's the bilirubin that's there as well. That's going to be used and introduced first into the small intestine at the duodenum, and it's going to attack fat. Right? One thing you should notice about fat, you try to wash it off your pans. It doesn't come off well because it's hydrophobic. But we can take that fat and break it into smaller little droplets, from big droplets to little droplets, because the charges inside the bile get in the way and help it to separate into those things. This, of course, is great because if they're separated into smaller things, that increases surface area and a greater chance of enzymes running into them. It's all about collisions. This, again, is going to be the pepperoni. It's going to be the cheese. It's going to be the Oreos filling. Oh, whipped fat. So tasty. Hey, that's lipids. Hey, lipase. Lipase is going to go after lipids. This is an enzyme, so this is going to take those fats and break them into their building blocks, which are, if you eat a lot of fats, you get fatty acids. So we got some serious biochem review here, right? This is when we're looking at things like triglycerides with a whole bunch of branching on that. This is going to break it into their single chains of those saturated and unsaturated fats, and those can work their way through a membrane because they're small enough now. This, again, will be the pepperoni, the cheese, all the good stuff that makes that grease, like you dip off the grease with the napkin, or you leave it on and just suck it up, right? Who's making lipase? Well, when in doubt, pancreas. If you don't know on the test, just say it's the pancreas producing it. Where's it going to be used? It's going to be used in the small intestine, introduced in the duodenum, right? Then there's this stuff called nuclease. Name tells you, hey, it goes after nucleic acids like DNA and RNA. And it's going to break them down into their building blocks called nucleotides, right? Lysine, guanine, ATGC, that makes DNA for me. Those break them down into the little rabbit-looking things, and those rabbit-looking nucleotides are now small enough to fit through a membrane. So that means that these long chains of DNA you couldn't get in, you can now use to make your own DNA because you are what you eat. That'll be the pepperoni, the cheese. That could be, of course, any of the veggies that are on there, anything that was a living thing. The crust will have it too because it came from wheat. This is produced by, well, find out, scrambled eggs. It's the pancreas doing it again. It's using the small intestine. So hopefully you notice this is mostly chemical digestion. It's all chemical digestion with these enzymes. But there's some mechanical digestion in the oral cavity with your teeth. There's some with the rugae inside the stomach grinding on that thing because it's a muscle. But then after that, it's basically the small intestine stealing the show. The large intestine does not show up on here because it's not used for digestion. It's just used to save water. It's a water reclaiming structure, right? Cool. Here's a way, of course, that you can actually read those. Hey, without all the bad jokes and commentary of guy repeating himself. Now, what we normally go into is looking at the other part of GR42, and the first part of that, of course, tackles things like the circulatory system, the cardiovascular system, which we'll start digging into this point. Here's some nice erythrocytes bumping along, carrying some oxygen for you, right? Bloody nice. Things that we should notice and put onto our GR. Why is the circulatory system important? When you have a health crisis, what does the ENT look at right away? The ENT comes along and he's like, oh, check the breathing, check the pulse, because that is how you get everything that's good from the outside into all your cells. The circulatory system basically takes the outer structure of your lungs, which is the outside of your body, or takes the outer structure of your things like your intestines, which is contact with your digestive tract, takes those good things and puts them in contact, gives them surface area to contact the rest of your body. So it's going to transport sugars, amino acids. It's going to drop off oxygen from the lungs. It's going to get rid of waste through things like the kidneys. And of course, it's your police force. It's traveling around there because it's connected to every single cell. Cells that don't have good blood flow, of course, tend to die really, really fast. But the fastest dying by far are brain cells. They can die within 1 to 2 minutes. That's why we always check the airway first, then check the heart. If you're having trouble with either of those, you're going to have problems, right? So if we look at the path of blood, that's good to know if you're going into medical school or for the AP. You can think of it kind of like as the heart is the engine that drives this, and the blood, of course, we could say starts, though it's a cycle, at the arteries because they go away from the heart. And the graphic that you can see here is that arteries are really, really thick-walled. They have smooth muscle. They can contract themselves, which allows you, of course, to take the heart and shove all that blood into there. And as you do that, it doesn't rupture the artery because it has all the pressure from the heart right away. Then they break into smaller things called arterioles, smaller arteries, and then they'll go to the rest of the cells through the capillaries, or the British, you call them capillaries. Now these guys are super thin. They don't have, of course, a whole lot of wall because their job is to allow diffusion to happen. And every part of your body basically has capillaries very, very close to it, except for like dense connective tissue, like your ligaments or your pads inside your knees. If we look at those pads, they don't heal very fast because they don't have a lot of blood flow because they're mostly just collagen, right? My meniscus. Then, of course, as the blood comes back, it's moving slow. It's lost the pressure of that. It goes through these things called venules. And then from the venules, it goes to the veins. And the veins always lead back to the heart for the most part. And if we look at the veins, those veins, their job is to bring it back towards the heart so that you can then shoot it back off to the lungs. As they're doing that, they're not as thick, they're not as muscular because there's not as much pressure, but they do have valves inside of them. Now, the crucial thing is that means that they're not really helping to get the blood back, so you need a little extra oomph for that. That's why a lot of your veins are arranged along your skeletal muscles, so that if you're sitting making videos for AP Bio or sitting on a plane or sitting in class too long or you've been vegging on YouTube, you need to get up and stand up every once in a while. That's why we take breaks so that you can have your skeletal muscles pump that blood back up to the heart because that's how you actually evolved to do that. Otherwise, you can get a clot in your lower extremities and that can travel to your brain. So you might want to pause me now and start walking around or just keep going. Now, in the old days, of course, in places like Scandinavia, if they wanted to know whether or not the son of a king had the right to take over the kinghood, what they do is they'd hack his arm, they'd chop his skin. And if the blood squirted out and he survived, that meant that he was strong because he had strong blood flow. If the blood just oozed out, well, then you were a weak potential king and they'd kill you. Now, the irony of this is, of course, when they hack them, if it's squirted out, that's because they cut an artery. And if you're an EMT and you show up and there's blood squirting out, that's a pretty good sign. You got to put pressure on it immediately because you're going to lose all of your couple quarts of blood very rapidly. And if you survive that, yeah, you must be pretty tough, actually. If it just oozed out, then that's just a vein, and that doesn't have as much pressure, so you don't lose blood as fast, but you still want to put pressure on it, right? Either way, unlucky to be the king. Now, a good example of a way that bodies manipulate it would be this beauty. This is an elephant seal. And if we look at the elephant seal, it's not too much to look at, but it is an awesome survival machine. This guy can dive 2 miles down to the bottom of the ocean and locate squid in the depths, and he can be underwater for 45 to 1 hour at a time. Now, I've tried, and I can last about 2 minutes underwater, but that's after a lot of training, and now actually with kids, probably just 1 minute at max. After that, I start to run out of oxygen, I black out, and I die. You would too. If we look at the elephant seal, how can it dive so deep? Well, the key is it controls its blood vessels. When it's getting ready for a dive, it will sit at the surface of the water for 1 to 2 hours, and it will sit there and it will breathe heavily. As it does that, it's loading oxygen into all of its myoglobin and its hemoglobin. That's the proteins that pick up oxygen inside of their muscles, the myoglobin, and also the hemoglobin in their blood. As they load up that oxygen, they then do a different thing. They've got all their blood in their extremities at that point getting warmed by the sun, loading up with oxygen. As they dive down, their capillaries in the outer part of their body, the extremities, actually constrict. And that causes the blood flow to increase to the inside of their body, but not to the outside. So as they dive deep and get cold, that cold doesn't get to things like their major organs. They also, of course, are storing all that blood with oxygen in, as well as the muscle oxygen, inside of crucial muscles to move down through that area, and their brain and their heart and their liver, so they can process those functions, find prey, and then come back up. It's a beautiful survival mechanism, that uses the cardiovascular system to modulate them. You also use this to some extent when you get cold and your skin gets pale because you're constricting the outer blood vessels and sending blood to your inner organs to keep yourself alive. So it has a variability with oxygen as well as heat. So if we look at the mammalian heart and circulation, we've got this cool heart with basically 2 fish hearts built into it. If we look at the cycle that's going on in that, we can cheat and say that it starts at the right ventricle, though it starts in the bone marrow technically, right? You're going to want to know the actual cycle of the heart. So you're going to start off here in the right ventricle. And when we look at these images, you have to imagine that the patient is looking at you. The right ventricle is the second biggest chamber. It's going to pump the blood to the lungs, and it's going to go through this thing called the pulmonary artery. As it goes into the pulmonary circuit, pulmonary being the lungs, it's going to travel in the lungs, drop off CO2, pick up oxygen with all those capillary beds and all that surface area. Then it's going to travel back to the heart in the only vein that carries oxygen-rich blood, the pulmonary vein. That's going to drop into number 4, which is the left atrium. Now, the atriums are small structures and thin because they've only got to squirt blood down into the ventricles. But the biggest chamber of your heart is the left ventricle, because that's going to pump not just the pulmonary lungs, it's going to pump through the aorta all the way to your systemic circuit. It's going to go up your carotid to feed into your brain and up to your brachial arteries. It's going to go down into your mesentery and your renal arteries to go to your things like your kidneys and your intestines. As it's doing that, it's dropping off the good stuff, the oxygen, the sugar. It's picking up the bad stuff like urea and CO2, and then it's got to go back to the heart. So when it does that, it travels through the superior and inferior vena cava. Those vena cavas, the big vein caves, are going to dump into the heart once again at the right atrium, and then boom, we're in the right ventricle. We start the cycle again using the lungs. So it's quite efficient in the system of picking up the good stuff, dropping out the bad stuff. How this evolves, of course, goes back to our ancestry. Before you left, we started looking at that. You basically can see that there's 2 fish hearts stuck together, and that gave us greater efficiency as mammals. This is different than, say, the open circulatory system of the invertebrates, like the crayfish that we looked at, or the grasshopper, or the spider that we've looked at in diagrams, where they're just squirting hemolymph, not even cells, and it's getting around their body. That works fine if you're small enough. That's why insects have a limit. They can only get it to the cells that are there. But when you develop a closed circulatory system, you can control the flow. And we can see what it does with the elephant seals. You can see what it does for worms and us, right? Now, the fish circulatory system has 1 circuit and basically 2 chambers. The ventricle does the same thing, pumps it to the gills, shoves it right there. That's why it's positioned there, picks up the oxygen, drops out the CO2. Then it loses its actual velocity and pressure so that when it goes to the rest of the body, it's moving really, really slow. That's why fish are cold-blooded. Then it goes through the systemic circuit, dropping off that good stuff, picking up the bad stuff, and it's got to make its way back. But it's lost a lot of that oomph as it's doing it. So that's why, of course, they're living in the slow lane. This, of course, tiles into the evolution and the steps of vertebrates, right? If we look at their circulatory systems, we can see how it's developed over time. Take a fish ancestor of all vertebrates and have them develop another chamber, and that would be the frog heart that you would see in your lab if we were doing it. Frogs have the classic Valentine's Day 3-chambered heart, and that 3-chambered heart means that they have a shared ventricle and 2 atria. In the shared ventricle, that means oxygen-rich and oxygen-poor blood are going to mix, and that's going to make their circulation less efficient. They interestingly have what's called the pulmocutaneous circuit because they not only send blood to their lungs to pick up oxygen, they also send it to their skin. That's why their skin has to stay moist so they can do, of course, diffusion. A dry membrane would cause them to suffocate. That's why you don't want to touch a frog's skin too much and you want to keep them in the water. Then, of course, it would come back to the heart, get mixed with some not so great low oxygen blood, go out to the system. If we look at reptiles, another step in evolution who evolved from the amphibians, they've got almost 2 separate circuits, but there's a little hole that actually allows blood that's oxygen poor to mix with the rich stuff. So they're slightly more efficient. That's why they can move a little better on land, but not completely efficient. Interesting things like crocodiles, they also have an additional vein that comes out, and that vein allows them to shut off flow to things like their extremities. That's why they can sit at the bottom of a pond for a couple hours, or if you're a turtle, for a whole winter, because they can reduce their metabolism being cold-blooded. Warm-blooded creatures can't do that. So mammals and birds, we've got to have oxygen supply all the time. We got to keep up our efficiency. We can't be inefficient like a fish. So we developed two separate circuits, and those are separated by a complete four-chambered heart. So you can pick up the good stuff, drop off the bad stuff, and do that without any of this doping of the two parts. But that means that we constantly need an oxygen supply, so we're always hungry. But that's why we've conquered so many environments that you wouldn't find reptiles or amphibians in, because we can, of course, supply that with the oxygen coming in and the ATP from our healthy diets. Now, normally what we'd go into is the frog dissection. You can find those online if you want. It's pretty sad that you don't get to actually see the pancreas, or, learn that you want to dissect with your mouth closed, especially because the gallbladder pops. But, ask some former students. If we look at frogs, things that we would have pointed out today, one, as you look at their skin is super thin. And it's super thin, and there's no fat underneath their skin because they breathe through their skin. If you flip over the skin, what you'll see is a whole bunch of capillaries, because that's basically like a second lung for them. So be very gentle with a living frog's skin. When we look at their lungs are actually pathetic. They're just these little sacs. And that's because, of course, they don't have a diaphragm. That's because they use a thing called positive pressure breathing, where their throat pouch pushes air into us. That's very different than our negative pressure breathing that we use. That's far more efficient, but that's why they have to breathe through their skin too. If you look at their heart, it's the classic three-chambered Valentine's Day heart with that doping in the middle of that shared ventricle. But it has a lot of features that look like ours because we're descended from amphibians as well. If you look at a frog kind of looks like a crocodile with its nose, its nares, and its eyes and its eardrum located right at the surface of the water, and some beautiful camouflage because he's a predator. He's also got to worry about birds coming down and killing him, so he's a prey as well. And that's why they've got that beautiful amphibian lifestyle where they can be at the merger, the line between aquatic and terrestrial. And you can look into their mouths, and what you'll see is Kermit has little tiny, basically vampire fangs. That's because he's a carnivore. That's part of the reason why his digestive tract is so darn short, and his liver is huge because he's got to break down the fats that are inside of those. And their tongue, famously, of course, can shoot out, but that's because their tongue is latched in the front, and they can reach forward with it, whereas ours is attached in the back. That gives them the extra reach they need because their arms are really, really small. And their arms have to be small because they evolved giant legs, and that would get in the way of them jumping. So a lot of cool features with them. One other thing that you would see inside is you'd see they have these fat bodies. The fat is inside of their actual abdominal cavity, not on the outside where the skin is, where we keep ours, because, well, that would prevent them from breathing. So they store their fat inside with their organs. We do that as well, but they don't store any of their skin. So no fat frogs. It's easy being green. Now, if we also look at what you'd be tackling after this, it would be GR42. You go after the rest of it. You'd be getting ready for a blood pressure and heart rate lab so you can understand things like systolic and diastolic. We'll talk about those in the next ETD. You got the 2016 that's coming up due on the 21st, and your self-score in the 2015. And I've just talked for way too long. So, hey, at least you got to pause me. I didn't get to. I hope you're doing well and staying healthy. Thanks for listening.